Today we have Dr. Kerry Nickols with us. She's actually just joined the Cal State Northridge program as an assistant professor. Prior to that, she did a double major in integrated biology and earth and planetary sciences at UC Berkeley before eventually pursuing her PhD in marine ecology at the Bodega Marine Laboratory through UC Davis. Her current research focuses primarily on coastal marine ecosystems like giant kelp forests, and she's really interested in coupling physical oceanography with ecology. With that, I'll turn it over to Dr. Kerry Nickols.
And I want to thank, Christina Frieder recommended me for the seminar, and then thanks to you for inviting me. And yeah, so I've only been in LA for a month. Are we okay over here? It's fine with me. Whatever you guys want. Yeah, so I've only been in LA for about a month, and before that I was a faculty member at CSUN Monterey Bay. So what I'm going to do today is I'm going to, since I'm new to the area and I want people here at USC to know who I am in case anyone wants to work together, I want to know more about Southern California too and systems here. So I'm going to kind of give you an overview of some of the different research projects going on in my lab. And one of the things that I think really inspires and drives a lot of us is thinking of the ocean as this like really heterogeneous place that has some really interesting features on a variety of spatial scales as well as temporal scales. And so if we see, so these are sea surface temperature images that were taken from the springtime. And so in the California current system, I'm sure a lot of you know that we have these times where we have upwelling and we have these cold water areas associated with headlands. And so you get these really interesting features that appear. If we zoom in on Southern California, the cold water coming off of Point Conception, that causes these Northern Channel Islands to be quite different from the Southern Channel Islands, and I know a lot of you do some work in Catalina and in this general area. So there's a lot of interesting features that can impact biology and ecology and drive distributions of organisms. And if this is, sort of what it looks like in spring, this image was taken from last week, so that same kind of area, but now looking at sea surface temperature now. And so there's also a lot of temporal change, temporal variability, and then of course we all know that the ocean is changing largely because of humans in some really profound ways. And one important question to ask that maybe we don't ask super often is, are there any signs of resilience by marine organisms to changes in climate? So we can ask the question, when organisms experienced past changes, were there examples of resistance or recovery? And maybe if we can learn something about the resilience of organisms and what led to the resilience, then maybe we can prioritize some management decisions as we're seeing, more and more change. There's not a lot out there in the literature about resilience of marine systems, but that doesn't necessarily mean that ecosystems haven't shown signs of resilience, and part of that is because we don't often publish things if they don't change. And so with some colleagues when I was at Hopkins Marine Station as a postdoc, we took an expert survey approach to try to get at whether or not maybe we've seen signs of resilience to climatic disturbances sort of in the past. And we were really focusing on whether or not some of these biogenic habitats, so kelp forests, coral reefs, mangroves, salt marshes, oyster reefs, and seagrasses had showed signs of resilience, so structure-forming species, habitat-forming species, if they had shown resilience to climatic disturbances. And so sometimes, if you're just observing a change, you wouldn't necessarily publish on it. So we sent this survey to 300 experts. So we picked the top 50 people that published, that had the most publications for each of these different ecosystem types. And we had about a 33% response rate. And so the way that this survey went about was that first we asked them about if they had specific personal experience, so if they had personal observation of resistance or recovery, which are sort of two parts of resilience, and asked, what type of disturbance that was, and then what sort of biological or physical factor contributed to the observed resilience. And then we also asked opinions. So this was sort of thinking of maybe not a specific, one example, but sort of a accumulated knowledge of an ecosystem, the factors that they thought promoted resilience or prevented resilience. And we sort of broke those up into resistance and recovery. And then the last section was we asked for literature examples in case, I don't know, we were just really terrible at using Web of Science and missed all of the papers, which wasn't the case. That was a joke. So then we asked them, are there papers that maybe we missed, and you could clue us into those different papers. So I'm just going to highlight a couple different results from those first two parts, A and B. So in regards to these expert examples, 69% of experts reported that they had observed resilience during their career, and of these, 80% witnessed resilience, specifically following an example of a climatic disturbance. And within a system that I work the most in, kelp forests, 92% of the people that responded, of those like 80% that did, observe resilience to climatic disturbance. So what, I think this part is maybe the more interesting for me is thinking of, okay, so you observed resilience, like maybe why did you see or not see resilience? So what can we learn about the factors that promote resilience? And so what we're looking at here, so these are factors that we had ranked and sort of grouped what people said into these different factors, and then showing for resistance in the white bars and recovery in the gray bars. And the ones that I really want to highlight are the two that had sort of the highest proportion of expert opinion saying that these promoted resilience were the physical setting of a particular ecosystem and habitat, and then also recruitment or connectivity. So, recruitment or connectivity would be like the amount of babies essentially entering a population. And that also is thinking about how connected different sites and habitats are. And both of these, physical setting and recruitment or connectivity, really depend on the oceanographic context of a particular habitat and the amount of environmental variability that is appearing. And so I work on a lot of different things because I get really interested in things that involve physical oceanography and ecology, but I think the sort of thread that weaves my projects together would be that I'm interested in how variable the environment is and how that influences the physical setting of a place, and also I'm really interested in recruitment or connectivity, which is highly influenced by the environment and the amount of variability in the environment. And then I think I'm trying to sort of form this new way of thinking about how that can tell us about ecosystem resilience. So I do a lot of work taking observations and looking at current conditions, and then I also do work that uses modeling to look at predictions of future changes and really to try to understand what's going on through a mechanistic context. So I'm going to focus this talk on 3 different sort of prongs of my research program, where I look at population connectivity in the coastal ocean, responses of species to marine protected areas, and then kelp forests in a changing environment. And a lot of the work that I'm sort of moving towards and the funding that I have is really in this sort of kelp forest area at this point. Okay, so most marine organisms, especially like if you're looking at coastal marine organisms, so a crab that's found in the intertidal, they move as adults, but that's not their primary dispersive phase. So hopefully you all know this, adult movement is not the greatest source of dispersal. That comes from the larval form, which can spend days to months, depending on the species, in the water column. They are often obligated to spend time in the water column. And so while they're in the water column, they are often dispersing and being transported by ocean currents. And so if we're interested in this crab population, say on the southern end of Monterey Bay, we may want to know, what are some of the different, where do these larvae go? Maybe they go north up towards Point Reyes. Maybe they go south towards the Big Sur coastline. Maybe some of them actually return back into the population from which they were released. Now the problem is that we can't tag larvae generally. We can't observe larvae as they're moving in the ocean, although there have been some really, exceptions of people, making some progress in looking at that kind of thing. But it's generally a difficult kind of impossible thing to do. But it's really important to know these pathways because that's going to tell us how connected different populations are along a shoreline. It also tells us, well, what happens if you lose this population due to overfishing or something? Can it be reseeded? What happens if we put a marine protected area in here? And sort of questions of resiliency of these populations can be tied to thinking about, where are the young coming from and what happens if they lose those different supplies, and is this population feeding other populations? And sort of the paradox of marine population connectivity has been that there's been a large body of knowledge that contributed to this paradigm that was that larvae are swept really great distances offshore because there are these tiny, tiny things and they're in this giant ocean that is very dynamic and is moving a lot. And especially in an upwelling system, there was evidence that when you have upwelling, so we have that strong winds blowing parallel to the coastline, moving from north to south, and that because of Coriolis pushes water offshore, and so we have water close-ish to shore moving then to replace that water. So the thought was that the larvae were all going offshore and being, swept way offshore, and there was some evidence for that. But there's also been a lot of recent evidence that suggests that larvae actually stay close to shore and to their natal site, and not just in the California current system, but in a lot of different marine systems all around the world. And so some of the work by Stephen Morgan up at the Bodega Marine Laboratory, so this is work that he did doing a transect going offshore from 1 kilometer offshore to 30 kilometers offshore. And these are different larval stages. So, these are like the younger larvae, these are the older larvae. And for all of these different stages, they're mostly found within just a few kilometers of the shoreline. And that was true even in times of upwelling and relaxation. So basically all across the board, they were found close to shore. And I think really it was that a lot of times we weren't looking this close to shore to see if the larvae were even there. So there seems to be this idea that there is a lot of retention. And this is true across species that have a variety of different behavioral characteristics. So behavior can certainly contribute to these patterns. But the thing that I was interested in was looking at the role of physical processes, which had been previously unexplored. And so thinking about if flow features close to shore could influence larval transport and dispersal and contribute to what we've observed here. And one of the things that happens is, so this is at Bodega, so we have some intertidal researchers here on the shore, and then the coastal nearshore mooring is about 1.5 kilometers offshore along the 30-meter isobath. And there weren't a lot of descriptions available about this body of water here and what water motion is like that and how that might influence larval transport. And this is a really important piece of water for a larvae if you're released from the shoreline. This is where you start and you end your life. If you're being infected offshore or moving offshore and then having to eventually come back close to shore to settle. And so I was really wondering if variability in currents in this region impact larval dispersal. And so the main questions were if nearshore currents follow some generalizable pattern that could lead to this increased retention that we've observed across a bunch of systems, and if then the currents could influence nearshore larval transport in a predictable way. So I deployed these instruments called acoustic Doppler current profilers at these different locations along the coast of California that had different bottom depth ranges and offshore extents depending on the bathymetry because the sites were very different from each other. And so this is kind of zooming in on one of the sites off of Pajaro, just north of Moss Landing in Monterey Bay. And so here's these instruments, and they use sonar technique to ping through the water column, and you get returns of looking at profiles of velocity throughout the water column. So I deployed them to these cross-shore transects, and really just looking at how velocity varies as a function of distance from shore. And if any of you are interested in sort of small-scale fluid dynamics, probably not as much as I am, but there are these things in nature, boundary layers. And so whenever you have fluid moving past the solid surface, you have zero velocity right at the wall, and then as you move away from the wall, you increase velocity. And so the idea here was that, well, maybe there's a coastal boundary layer because we have water is getting shallower, and you also have this coastal wall. And so it would be reasonable to assume that maybe there's different patterns and velocity as you move away towards shore, and these lower velocity areas that could have an appreciable impact on larval dispersal. Okay, so I'm going to show you some data where we're looking at shores here, and then we're looking at along shore is the component that's parallel to shore, and then cross shore is the component that's perpendicular to shore. And then I'm going to step through instruments. This is one that was like furthest away, and then we're going to go closer to shore. And the velocities I'm showing you here are depth average velocities. So the first point is that velocity is strongest in the along-shore direction. So we say that it's polarized in the along-shore direction. So most of the motion close to shore, and this is nothing new, most of the motion of water velocity close to shore is moving this way and not this way. So here's the cross-shore velocity component is in this dashed line here. So if we look at the instrument that's like next closest to shore, you see that the mean as well as the amplitude of the oscillations of velocity increase as you go towards shore. And then again with this more inshore instrument, and then with the instrument that was closest to shore at this site. So these particular data were for a site that was just off of Santa Barbara area, but now I'm going to show you data for all of the sites. And the way that I've chosen to compare data across sites is really plotting them like these small scale boundary layers. And so the way that we plot that is to look at distance from shore here on the x-axis, and then we're looking at velocity on the y-axis. And I'll plot these different sites. And in order to do this, we're not looking at a time series anymore. We're also looking at not only depth average velocities, but time averaged velocities. And they're time averaged over a week because that is a period of time that's relevant to dispersal of some larvae that don't disperse a whole lot. That would be things like abalone, mussels, oysters, but weeds, things like that. So here, this is the site that was off of Santa Barbara, and so seeing that slower velocity closer to the shore and then increasing as you move away. This is the site Pajaro, that's just north of Moss Landing in Monterey Bay. Again, see that similar pattern. Here's Huntington Beach, Hopkins Marine Station, and then Terrace Point was just off of Santa Cruz. It's hard to see, but there's a data point here, so it sort of goes out then. Some of these are kind of asymptotic at the end. So again, we were thinking that maybe this is going to look like sort of a boundary layer, but it was never expected necessarily, and especially these sites have very different topographies, like how you see that, this site is going out to 30 meters or so around here, but has a very steep shelf. So some of these sites have steep shelves versus broad shelves. And so to see what's the relationship between these different, between distance from shore and along-shore velocity, to see if they really are similar to boundary layers. The way that people look at boundary layers is to, there's this law called the law of the wall. So this is a universal relationship between velocity and distance from a wall that has been studied across a whole bunch of different types of bottoms, so like over different rocky substrates, and looking at mostly like flow through pipes and things like that is how fluid dynamics has looked at these boundary layers. But there's this universal relationship where velocity is related to distance from a wall, and it's a logarithmic relationship. I'm not going to get into details of these, but if you're interested in them, I can talk to you about them. And so the question was, are these boundary layers that we're seeing logarithmic as well? And so if you plot them in this way, indeed they do kind of collapse all to this line. And so it does appear that these coastal boundary layers, the physics are different, and I can go into, so I wasn't going to talk about really the mechanism that's driving this, but it is very similar to a classical sort of small scale hydrodynamic boundary layer. And that's, I think, really interesting because, again, these sites are from different places along the coast. They have very different shelves, but yet at all of these 5 different sites across California, we see this pattern. So here's just a schematic of how, the next time you look out at the ocean, maybe if you look past the waves, we're not talking about the waves, we're talking about things just past the waves, that there's this region of slower velocity that increases as you move away from shore. And people, I guess I should have backed up and talked about this a little bit earlier, but that area between, generally where this mooring is and where these people are, now hydrodynamic models have gotten better, and so we have smaller scale resolution. But there was a very large period of time where the closest sort of model cell for velocity that you had to shore was at least 2 kilometers away from shore. And so all of this variability was collapsed into a single value. And now we have these higher resolution hydrodynamic models, but they're very computationally expensive, and you have to nest them inside these other models. And so you can't just like figure out what, that level of resolution and velocity is for any area in the ocean necessarily. But the point here was to see, okay, does this pattern exist? And then to go a step further and ask, well, would this matter and would this change our descriptions of larval dispersal? One of the ways that we look at larval dispersal is to use a thing called a dispersal kernel. These are just hypothetical ones that I've plotted up here. And what we're looking at, so if we say release some particles here in the ocean, how far do they go along the shore? So on the x-axis is the along shore position, and then on the y-axis is the probability of settlement. So we release larvae, and then how many of them come back to shore, and where do they end up going? So we can look at things like mean dispersal distance. So the mean dispersal distance of, say, a population represented by this kernel would be, about 60, and for this population it'd be about 140 kilometers. Another thing that we can ask is the amount of self-retention, and this is important if you're thinking like how many babies come back to home, and the way that we're defining it here is the fraction of released larvae that return within 10 kilometers of the release site. So this particular population would have about a 3% self-retention. This population here would be less than 0.1% self-retention. This gets really important when we're thinking about marine protected areas along a shoreline, and again, thinking of how connected different populations are. We can also look at long distance dispersal. So how far are the ones in the tails going? So what I did is I used a particle tracking model and incorporated this coastal boundary layer into the particle tracking model, and then compared results thinking about whether or not you include a coastal boundary layer, or if you're just looking at sort of like business as usual, like velocity is all the same at the near shore. And so the particles would start out at a certain point. They'd have a certain along-shore velocity and cross-shore velocity that came from my observations that I made in the field, as well as an along-shore diffusivity and cross-shore diffusivity. So this is talking about sort of spread of particles that also came from the velocity measurements that I made in the field. And so you end up with something that would look like this if you were to look at an individual track of a particle that say was like released here. And so this was one that didn't have a coastal boundary layer in the model, and so this is along shore distance and cross shore distance. You can see it's traveling, it's meandering, there's a random component to the random impulse in the model. And then this is one that ended up settling that had a coastal boundary layer included in the model. So of course they didn't just look at like 1 or 2 particles, they looked at tens of thousands of particles released into a model field, and then aggregate the results into those kernels that I showed you. So here's for 2 different sites. So Mohawk that had really high velocities and a really steep shelf versus Pajaro that had lower velocities and a broader shelf. And if particles are released here at the 0, and then we let the larvae do their thing, and then look at it after the one example I'm showing you here is for 10 days. We looked at them over a variety of different pelagic larval durations. And the thing that first I want to point out is that, so this is without a coastal boundary layer in the model. And the reason you see this big peak right away is because, so most larvae have a stage where they are pre-competent. They're not competent to settle, so they're obligated to be in the water column. And so then once that competency window, like the pre-competency window ends, and then they're able to settle, you can often see like a big settlement peak in the model. So that's what's happening kind of like there, like one aspect of that. And a lot of models that use hydrodynamics and then look at larvae don't often include some details of the biology of the larvae, and so the pre-competency window is often missing from some of the larval dispersal models. And so here's a kernel that results with having a coastal boundary layer in the model, and so you see that you have, for both of these different sites, that you have lower mean dispersal distance. you have a higher chance of self-retention, more staying closer to the release point. But also you can still have long distance dispersal. So the tails, maybe are a little bit truncated, but like you still can have, it doesn't preclude long distance dispersal. It's just explaining the fact that you might have increased retention closer to shore with these, incorporating this idea that there's lower velocities closer to shore. So this is looking at 2 different pelagic larval durations at 2 different sites. But if we want to look at all of the sites for all the range of pelagic larval durations from the model, that's what's shown here. And then now what's plotted on the y-axis is the ratio of the mean dispersal distance with a coastal boundary layer to that without a coastal boundary layer. So if you're up in this area of the plot, it means that the coastal boundary layer didn't really have an effect. And then if you're down here, the coastal boundary layer is more important. So the coastal boundary layer is more important to include in these descriptions for lower pelagic larval durations, and also these sites that have broader shelves. But, even out to a 30-day pelagic larval duration, there's still, about a 10% difference that can be made by incorporating these flows closer to shore. If we look at self-retention, that's what's shown here. So we have pelagic larval duration on the x-axis, and then I just highlighted this 2 of the different sites. And then on the y-axis is the fraction of larvae that are self-retained. So if it was 10 to the 0, that would be 1. That would mean all of the larvae were self-retained. And this is without a coastal boundary layer in white bars, and then with a coastal boundary layer in black bars. So without a coastal boundary layer, we only really saw self-retention at this one site, Hopkins, and there wasn't any expected for Terrace Point. But once you include a coastal boundary layer, you actually do get a lot of self-retention and out to a pelagic larval duration of 12 days. So to sort of corroborate this work looking at what's going on in the near shore, I wanted to take it a little step further and make sure that there actually are larvae that close to shore. And so where Stephen Morgan's work that I showed you earlier ended here at about 30 meters, about 1,100 meters, 30 meters water depth, 1,100 meters from shore, I then did some work looking further inshore to see, are there still larvae in this area where we're observing these lower flows. And so what we're looking at here are crustacean larval abundance, and there was a lot of larvae found within this area close to shore. It seemed to peak around 22 meters. This transect was done off the coast of Bodega, and I went out about 6 or 7 different times under conditions of both upwelling and relaxation. Again, saw larvae under all conditions. And if we break that down into the different larval stages, so here's looking at barnacles and here's looking at crabs, all larval stages are found within this area 1 kilometer from shore. It's interesting that we see higher abundances at these deeper depths, and I suspect that might have something to do with getting closer and closer to the surf zone, which then is a whole other crazy area that I don't work on, but Stephen Morgan does some really great work on looking at larvae in the surf zone. So this is probably something I'd want to follow up on, because the very, very near shore and these really shallow depths do seem to maybe be a little bit different for larvae than the ones further offshore. So just to sort of recap, that variability in currents can impact larval dispersal. So this feature called the coastal boundary layer of reduced flows close to shore decreases predicted dispersal distance and also increases self-retention. And this is a physical mechanism that corroborates studies that show high self-retention. So, then you think we already have these flows close to shore that are happening physically that are decreased, that can increase retention. So if you add larval behavior on top of that, you're probably going to see even, higher increased potential retention. Okay, so switching gears a lot. I'm going to tell you now about some work looking at responses of species to marine protected areas. So marine protected areas are a popular management tool. They're used all across the globe. And in general, when people put in marine protected areas, there is an expectation a lot of times to see a positive change in some biological measure. So here we're looking at biomass, density, size, or species richness. And for a lot of marine protected areas, that is what you do see. You see increases in some of these different parameters. But there are times when you don't see that, where you actually see decreases in biomass, density, and size. And so a question is, do marine protected areas always meet their goals, and why or why not might they not meet their goals? Hopefully you realize that California has the second largest network of marine protected areas in the world. California is an awesome place for marine protected areas. They are generally pretty small, but we have a lot of them. And so here are the MPAs prior to 2003, and then the MPAs effective in 2013 as part of the Marine Life Protection Act. And the red dots are the different MPAs, and then these squiggly lines are just showing you these different regions, because that's how they were implemented, is that the state went to these different regions and worked with stakeholders to design this network of marine reserves. And it was mandated by law that they were to be a network of marine reserves. And they're not all reserves, marine protected areas. Marine reserves mean totally no take, but some protected areas do allow take. Some of these are marine reserves though. So when they went in the central California coast, so that was the area that was implemented first. They went in 2007. And 5 years later, there was this big meeting in the state of California Central Coast, and people looked at some of the monitoring data and looked at plots like these, which are showing the relative increase in biomass or a relative decrease in biomass. So if you were on this line, there's no change. So people really want to know, okay, it's been 5 years, are these things working? Are the MPAs meeting their goals? Should we have MPAs? Like, what's going on? And this can be kind of a difficult question to ask because we're looking at all of these different types of species, and the question is, should they be meeting the goals? Should they be increasing? But it's hard to know like what the expectations are unless you really know about sort of the population dynamics of these different fish. So I work with some people to use population models to understand what happens to fish in marine protected areas. And so specifically we use what we know about the age and growth of fish, about larval recruitment, about fishing rate. We put that into a mathematical model of fish population change to make predictions about how much change there should or could be after a marine protected area, as well as how fast that change might occur. And then we look at monitoring data, so actual data from those different MPAs, and we use that monitoring data to actually fine-tune our mathematical model of change of fish populations. And then we can also update some things that we know as we tend to know more about those parameters too, about the history of the organism and things about how much fishing was happening before these MPAs went in. And really we're trying to understand what influences the responses of different populations to marine protected areas. So we've been working with these 3 different fish species. Today I'm just going to talk about blue rockfish. And all of this data comes from this kelp forest survey data from the PISCO group, the Partnership for Interdisciplinary Studies of Coastal Oceans. If you're at all interested in any data related to marine protected areas, PISCO has data that's publicly available. And they're really some of the main people that are monitoring these changes inside these marine protected areas. And then we look at these sort of different regions of marine protected areas within the Central Coast. So the area around Point Lobos, which is in the southern end of Monterey Bay, Big Creek, which is along the Big Sur coastline, and then White Rock, which is closer to Cambria, sort of north of San Luis Obispo. And I'm going to highlight first some results from Point Lobos. So just to kind of put this in context about this issue about trying to understand what's happening. So here we have a time series. So this is time on the x-axis, and then how many fish you have. And really we're focusing on the fish that are big enough to be caught on the y-axis. So here's 2007 when the new MPAs went in. So the expectations generally when you put in an MPA, so there's also some old reserves in this area too. So if this was the amount of fish that you had outside of your MPA, you might expect we don't have fishing going on, so we expect to see higher fish abundance inside an MPA. When a new MPA goes in here in 2007, the expectation would then be that it would increase after some time to the same abundance as the older reserve. So is this what we see when we look at blue rockfish data? Oh, I'm ahead of myself. Okay, so pretend that that didn't happen. So this is the actual data. So here's what we're seeing. So these are the data inside. So you could kind of say, okay, like the old reserve, like maybe has more fish, but there are some years where definitely like there's not much of a difference between inside and outside the reserves. And then after the new reserve went in, like, whoa boy, all the fish were doing pretty bad for some reason, starting in, about like 2007, we put in the new reserve, like maybe by 2010 it was increasing, but the data are really messy. And it's hard to really understand what's happening. And so, people might look at these data, especially from a context like if you weren't excited that there were marine protected areas, you might say the MPA is failing, we don't need the MPA, I don't know. Maybe it takes more time to see an effect, so maybe there hasn't been enough time that's occurred, or perhaps there's also too much noise, is another reasonable assumption to make. So when we're trying to figure out what's going on with these marine protected areas, one of the issues is that the monitoring data are coming from these individual sites within an MPA. But what we know about the amount of fishing that has occurred is for stock assessments that provide a value of harvest rate or fishing mortality rate for the entire California coast. So you can imagine that there might be differences between where you are in California and how much fishing is going on. And so there's this rate was from the stock assessment of the fishing mortality rate for the state of California for this species. But you'll notice too that if you're here in the Point Lobos area, you're a lot closer to human population centers, you're really close to Monterey Harbor, so you might have a different impact. So these arrows are showing you where some of these different fishing harbors are. Yeah, there's some fishing harbors in here, there's also a lot less people in San Luis Obispo County. And so, in Big Sur is really hard to get to. There's not a lot of fishing ports going on down there. So you might imagine that the amount of fishing occurring is going to change depending on where you are. So within this Point Lobos area, for instance, these are all different sites where the monitoring data comes from. Here is where the old reserve was, and then in 2007 it was expanded to include this amount of area. So, using a fishing mortality rate for the entire state might not necessarily apply to these different places. So what we do is we estimate demographic parameters like the fishing mortality rate by fitting a model to a time series of size abundance survey data. I use a Bayesian state space integral projection model, which I'm not going to get into, but if that's something that you're interested in, I'm more than happy to talk to you about it. And then what we can do is use estimates of these demographic parameters to then simulate expected trajectories of populations inside marine protected areas. And we can then compare it to data too, and see how quickly we're matching our model to the data. So this is sort of the modeling framework where you have a model estimate of a population state. You include things like fishing and recruitment and process error to get an estimate of that next state. And then you can compare that, so then you actually realize that, oh, maybe that isn't exactly what you observed, and then you compare that to the data, and then that goes back into having new estimates. And so it's this Bayesian approach we're using maximum likelihood. So this was the prior fishing mortality rate, the Bayesian prior from that statewide stock assessment. Once we looked at the model estimates of the actual fishing mortality rate based on the monitoring data that come from these different areas, the fishing mortality rate for the Point Lobos area was estimated to be over twice that of the Bayesian prior. The area around Big Creek, the fishing mortality rate was close to 0. And then the area kind of around Cambria is actually pretty similar to the statewide fishing mortality rate. So that's one thing that's going to contribute to these differences that we're seeing in how populations are responding to marine protected areas is actually like how much fishing was going on. If you put in an MPA somewhere where there was no fishing going on, you're not going to expect to see a change. So another complication, especially with rockfish species, are that their recruitment is super variable. So if you look at a time series of recruitment from the Point Lobos area, there are some years where there's high recruitment. These are looking across a few different sites. That's where the error bars come from. There are also years where there are total recruitment failures. So some of you in the room may remember back in about like 2005, 2006, that there was just crazy ocean conditions. There were a lot of seabirds dying. Things were not good in the California current, and that corresponded to a time of really low recruitment for these rockfish. And so it's important to think about that, like, when we're looking at responses of species that they don't have constant recruitment, that a lot of times the number of young coming into a population is really variable and is often related to ocean conditions. If you just look at a model that has constant recruitment, so like there's zero variability in recruitment, then you expect to see like that really simplistic thing that I showed you before, where this is time after MPA implementation. This is with fishing, so if we had a fished population versus an unfished population, if you put a reserve in, the number of fish increases to that unfished population state. But this is assuming we don't have variable recruitment. If you incorporate recruitment variability, then you end up, you can, so what I do is I add recruitment in sort of randomly, and then I look at the sort of envelope of outcomes of a bunch of different model runs. So that's what's showing here. So now we've got variability around this fish population. This is the mean of the model runs, and then these are within the 95% of the values from the different model runs. So a fish population, put in a reserve and it increases. And you see some difference between the reserve and the fish population if you have, a measurable amount of fishing that occurred. But there is this overlap that occurs for about 10 years where you wouldn't be able to tell the difference between a fish population and a reserve population because of the recruitment variability. But we also know that our fish populations in our marine protected areas probably didn't start out at this like magical equilibrium population size. And especially if you look back at this recruitment time series and realize that those marine protected areas went in 2007 at a time when there was like this crazy amount of low recruitment. So that's going to be a problem kind of going forward. And so what we're looking at here is starting a model from the 2007 abundance of fish and then incorporating variable recruitment into the model. And again, the solid line is going to be the median of the model runs, and then these dashed lines are the 95 and 5% sort of envelopes of all of the different model runs. And this plot should look confusing because basically you don't see a difference for looking at Point Lobos, that site that had the highest amount of fishing mortality. Even after about 20 years, you can't tell the difference between a fished population and a reserve population. So it has to do with the recruitment variability, the fact that there was really low abundances sort of when the MPAs went in. And this matches up to data. So these are the actual monitoring data that fall within sort of expected outcomes in the shape too. And so this dip that you see in the data and in the model are because you had those like a really low amount of babies, and then that then sort of like cascades through the population size structure as time goes forward. At Big Creek, where there was like no fishing mortality rate, there's no difference between a reserve and not reserve because there's essentially no fishing mortality. Looking at the data for this one is a little bit crazier because they only went out for 2 years and there's a ton of variability, especially like this one. Yeah, they had they sample a lot fewer sites because the site is harder to get to as well. So why are blue rockfish more abundant in marine protected areas? Variability in recruitment that's driven by ocean conditions hampers the ability to detect these responses of fish species. And then also this idea that there were low recruitment years prior to implementation also impacts the response of species to marine protected areas. And so really what we try to press upon people, especially when we talk with people from state agencies and decision makers and managers, is that you have to have expectations in order to be able to evaluate marine protected areas. And that models are a really useful tool to help develop expectations. And also a way to interpret the monitoring data. It's like a mechanistic way to interpret the monitoring data, especially in cases where we don't really know the fishing mortality rate and we need local estimates as well as recruitment variability. So thinking about the timing and life history of the different organisms. And also models can then inform the design of the monitoring programs and adaptive management. So to make sure that we're even like collecting the right monitoring data, models can be a really good tool for that. Okay, so then I'll just touch on a little bit this sort of last thing that this is the direction that a lot of my work is going now, is thining about kelp forests and the fact that they're in this very dynamic ocean environment. So kelp forests are foundation species. They provide habitat for ecologically and economically important species. They house large amounts of biodiversity, and they're very productive. And I'll just add that pretty much everything I know about kelp forests mainly comes from Central California, and I'm gonna have to relearn everything now that I'm in Southern California because it's different here, but I'm excited to learn. So kelp forests are also, experiencing climate change. We have changing temperatures, increased storms, increased upwelling in some areas, as well as sea level rise. And then also ocean acidification is, of course, going on. And people are really interested in ocean acidification, especially in Central California, where there is a lot of upwelling, and those upwelled waters are high in carbon dioxide and can impact the organisms that you see here in the benthos. And people are also looking to kelp as kelp and seagrasses as these habitats where you could change the water chemistry. And so people are, like state agencies, are really wanting to know if this is potentially a mitigation strategy, but there's very little known about the biogeochemistry of the kelp forest environment. Christina Frieder, your wonderful postdoc that's here, has done some work on this question, and so this is sort of taking some of those questions that she started asking kind of to the next step. So, for instance, it's also complicated because there's a lot of different things going on. So we have anthropogenic CO2 that leads to decreases in pH and increases in PCO2. Upwelling, cold waters hold more gas, so they're more rich in carbon dioxide, so we see decreases in pH and increases in CO2. And then respiration, of course, is going on, which has decreases in pH and increases in PCO2. But then there's primary production, which can increase pH and decrease PCO2. And so you can't just look out the kelp forest and know exactly what's going to happen, because you have to kind of know what's going on within these different sort of pools of changes to the carbon system. And so the questions that I've been starting to look at and will continue to look at are what patterns in water column chemistry are associated with kelp forests, and does the physical setting of the kelp forest influence these patterns? And we know that the physical setting can impact kelp physiology, so there's been a lot of work that's looked at how water motion can influence uptake rates, different things like that. And then, of course, the kelp forest also, and I think this piece sometimes gets left out, impacts the physical setting itself. So it turns into a very interesting system to work on. So how, the kelp forest can influence, individual kelps can influence turbulent mixing, that can influence the amount of damping that the canopy does of the currents. Those can affect the influence of surface gravity waves as well as internal waves. If you look at the whole forest, a variety of studies have shown current attenuation within a bed, so lower flows inside a bed, but then you get acceleration along the outside of beds. And you can think of this if you've ever seen flow moving past a rock, right? It hits the rock, and so then it has to go around. Well, the kelp isn't a rock, but it has some solid structure to it. So you're going to get some current slowing down in the kelp and then redirection of flow around the edges. And there's not very many studies that have looked at attenuation of velocities in the kelp, but the few that are available, follow this relationship where as you increase kelp density and distance into the center of the forest, you get greater amounts of attenuation. And then something we don't really know very much about is how that influences sort of exchange between beds and the fact that they're also embedded in that coastal boundary layer with lower flows closer to shore. So I did some work this is Hopkins Marine Station in the southern end of Monterey Bay. This is Stanford's Marine Lab, if you haven't heard of it before. And this is a freakishly calm day there, but I swear that this is the wave exposed side, and this is the wave protected side. And then there's this area here, the kelp forest array, that has a cabled observatory to it. And so for 14 months, I collected a weekly time series where we measured some carbon system parameters at these different locations and also did CTE casts to get the hydrographic properties. And then I also had current meters located inside and outside here and inside and outside on the wave protected side. And we took samples around the same time of day to try to sort of standardize something. And the parameters that we looked at again, so water velocity, temperature, salinity, DIC, and TA. For the DIC and TA, we were taking water samples 1 meter below the surface and then 1 meter above the bottom. And then from these parameters, we could then calculate PCO2, pH, and aragonite saturation state. I'm going to present PCO2 data because it's something that people tend to be able to wrap their heads around if you aren't used to thinking about some of the other nervous system parameters. It's important to note that, if you want to measure these things, or if you want to estimate these things, that you have to measure at least 2 parameters of the carbon system. And these tend to be the ones that we have the most precision. And this work was in collaboration with Rob Dunbar, who's a biogeochemist at Stanford and has some really amazing analytical facilities to look at these different parameters. So over time, unlike in Southern California, we have times where the kelp canopy gets ripped up by winter storms. And so if we look at kelp biomass over time, so this is the wave exposed and wave protected, these estimates are from satellite imagery. So this part of the project was done by Tom Bell, who's now at UCLA. And so, you see this typical sort of seasonal cycle where you see decreases in kelp canopy biomass, and then it starts increasing in spring and also during alkaline time, high nutrients, and then it starts to decrease towards the fall. Now I'm going to show you some of the PCO2 data. So this will look here first at the surface data, and we're looking at inside the kelp forest in the open circles, and then offshore of the kelp forest in the filled-in circles, and looking through time. So July, so this is the whole time series, and these are monthly averages, or averages across each month. So the first thing that you'll see is that at the surface, inside and outside the kelp forest is really pretty similar. And this I've shown, here's 400 microatmospheres, and so you definitely see that during the winter time that the PCO2 increases when you don't have a lot of biomass in that surface happening in the surface. At the bottom, there is more of a difference between inside and outside the kelp forest. And so there are lower amounts of PCO2 inside the kelp forest than outside the kelp forest. But during winter time, when there really isn't any kelp, those values converge between inside and outside. If you go over to the more wave-exposed site, so it's the same sort of setup, so 1 meter below surface, again, no difference inside and outside the kelp forest at the surface. And also at the bottom, no difference between inside and outside the kelp on the wave exposed side, as opposed again to looking at the wave protected side, where you do see a pretty big difference between inside and outside when that kelp is there. I think probably what is happening at the surface is that there's so much sloshing and things happening from the surface waves. Also, we weren't really looking at phytoplankton, which is another thing that we're going to start kind of looking at in the future, of how those contribute to the patterns. But I think what this story really shows is maybe not so much the influence of kelp, but the influence of infection in these different systems. And so what we're looking at here, this crazy spiralogram things you had when you were a kid, the circles. Each circle is a week of velocity records, and it's a principal component axis. And so the ellipse is showing you the axes of maximum variability. And we're looking at an ADCP in blue, so a current meter that was on the wave exposed side inside the kelp forest, and then a wave protected side inside the kelp forest. And so the predominant way that water is moving on the wave exposed side is in this axis, which is going inside and outside the kelp forest. So there's a lot of flushing going on that more wave exposed side. Whereas on the wave protected side, most of the motion is going this way, although it's pretty similar between both of those, and that aligns with sort of this axis, so sort of sloshing back inside the kelp forest. And these are some things that I'll be looking at in a more mechanistic way, in more detail, but these are just sort of the general patterns that we observed. So, people are wanting to know, can giant kelp be used to mitigate ocean acidification? And I think, really the ability of giant kelp to alter the water column chemistry is a function of the physical environment, which kind of goes back to the whole the sort of setup that I provided at the beginning. And people are answering, trying to ask similar questions in seagrass, and working with some people doing some modeling, trying to get at this question. And there's a lot of field data being collected, especially by the Data Marine Laboratory, going to different seagrass beds and asking kind of similar questions. But you really have to combine the physical data with the biological data and biogeochemical data to try to get at this. And then I'm collaborating with the Bay Foundation on some work looking at, they're doing some kelp forest restoration, and so we're looking at how the hydrodynamics of the kelp forest change as the kelp is growing in, and then we'll start looking at some of the biogeochemical questions. I will be returning to Monterey over the next couple summers with funding from NSF to look more in depth at these patterns that I observed. So if you know anybody who's interested in a postdoc or in getting a master's degree, then talk to me or send them my way, because I'm looking for people to join my lab at this point. So those are my main three things that I wanted to share with you today. And again, sort of this key theme of being that environmental variability can influence the physical setting and recruitment and connectivity, which can influence like what we're seeing currently, as well as the future changes, and hopefully can give some insight into ecosystem resilience. Here's some of the co-authors on the work that I was talking about today, and then a bunch of other people who have helped out with a lot of the different field projects that I've worked on. So thank you so much for your time.
Kerry?
Yeah.
Well, first, a personal comment. Your shout out to Christina Frieder. She couldn't be here today. She had a baby yesterday.
Yes.
So
Figured that.
healthy mom, healthy daughter. So
excellent.
Very proud, very pleased about that. NPAs. I was wondering they're so hard to work on, as you so well say, with variability. So let's look at, say, for example, what would we learn from terrestrial marine environments? Thinking about, for example, when various issues are protected and we prevent hunting, then they still have to go out and cull things and kill the wolves and kill the moose. The whole thing just seems to be in turmoil as to, even in those systems where you can count stuff a lot easier, right? So I'm just wondering, maybe some broader view, not, for all of us, right? Thinking about whether these ideas work at all. It's pretty shocking that your models tell us we have to wait 20 years to see if it'll work. Right. Nope, I don't think society will have that patience when it comes to stopping fishing, right?
Yeah, absolutely, and especially because of the fish here, like rockfish are, I also forgot to mention this, like rockfish are really long-lived fish. So think about that when you eat your rockfish that is probably older than you.
Yeah.
So, and there really isn't, like, when we think about, we're not like killing a lot of, like, we don't eat when we eat fish in the ocean, we're often eating things that are really old, which doesn't necessarily happen in the terrestrial system. So first of all, it's crazy that we do that. And we kind of have to take a step back and think, like, people look at that monitoring data and they want to see increases, but MPAs do a lot of other things, like, restoring the system back to maybe sort of closer to what it looked like before we started removing things from that system. And that it has to do with biodiversity. And there's been a lot of great work by Theo McKelly's lab at Stanford showing that at these MPAs in Baja California, there was this crazy hypoxic event that went through that killed off a lot of the abalone in these fishing areas in Baja. And in the areas where they had MPAs, that's where you saw the populations bounce back. So we need to, I think, look at MPAs more as like a, like putting money in the bank or something, I guess. It's like a terrible analogy, but maybe it kind of works. But it's like setting aside something. It's not going to be the cure-all to like all of our problems about fishing, and sort of moving away from thinking like, oh, I need to see like all of these things increasing. Yeah, it is a complicated thing. Yeah.
I wonder, do you look at the size distribution of the catchable fish? You get bigger fish in the MPA, not more, but bigger ones?
Yeah, so that's the expectation, but if you look at the actual monitoring data, especially for that species, you don't see that.
You don't see that.
And so, so that is another part of it too. It's like, okay, well, why aren't we seeing the big fish? And part of it is sort of the thing that I was showing. But the other thing too is that A lot of these species, they're not isolated in the kelp forest, right? They might have some sort of ontogenetic migration. So I didn't go into the details, but the model actually includes an ontogenetic migration to account for the fact that some of those species do move offshore. But the only reason that we even know that that happens is because there was monitoring data from inside the kelp forest and then a totally separate effort doing monitoring data further offshore doing hook and line fishing. And so being able to combine those monitoring data sets. But it's hard enough to get one long-term monitoring project funded, let alone like, measuring them in 2 different areas. So it complicates it, yeah. I think there was a question in the back, yeah.
When they set up the marine protected areas, did they consider your boundary layer issues or not?
I don't think they did because I hadn't finished it yet. Yeah. But they did think about larval dispersal. There were coastal oceanographers that were working on the science advisory team for the MLPA. So they thought about connectivity, but I think they were also, it's also hard to think, you're putting in MPAs for all of the species, and then people want to know, well, how are the populations connected, but how do you even decide, like, which populations do you look at? Like, you're not going to make a model for everything that's going on, so, yeah.
You mentioned that you're going to start considering phytoplankton in some of your future kelp forest work. I was wondering if you could just give us a sneak peek about some of what you're thinking going into that.
I'm going to take a net, put it in the water, and so I also have a fluorometer on my CTD, but I'm hoping that a component of it could be to look at community composition and to look at how that changes like over time and inside and outside the kelp forest. Because I imagine that there are like different players, but I have no idea.
All right, let's thank
Okay,
Kerry.
thank you.